Hey everyone, and welcome. Today, we are getting into one of the most fascinating and, honestly, challenging organisms you could ever come across at the lab bench. We're talking about a bug that basically throws the rulebook out the window and forces us to rethink our classic ID algorithms.
So, let's just jump right in with a question that, to any microbiologist, sounds like a complete contradiction. A Staphylococcus that has to have anaerobic conditions to grow, and it's catalase-negative? It just feels wrong, right? But that is the exact profile of the organism we're breaking down today.
The organism at the heart of this puzzle is Staphylococcus saccharolyticus. It's earned its nickname as a great mimic for very good reasons, and today we're pulling back the curtain on its unique biology, why it matters clinically, and how it's managed. And just so you know, all the info in this explainer comes from a comprehensive Gemini deep search. You can find all the primary sources listed in the notebook document linked for you. All right, so let's get into it, and we'll start right where every microbiologist does, at the bench. How in the world do you unmask an organism that seems perfectly designed to slip right past our standard protocols? You know, the diagnostic journey for Staphylococcus saccharolyticus is like navigating a minefield. It starts out simple enough. You do a gram stain, you see these classic gram-positive cocci and grape-like clusters, and your brain is just screaming Staphylococcus. But then, things get weird. You check your standard aerobic plates after 48 hours, and there's nothing. At this point, the culture could easily be tossed as negative. But if you happen to set up an anaerobic plate, and you're really patient, you might see these tiny little colonies pop up after 4, 5, maybe even 7 days. And here's the final kicker. You go to do a catalase test, and it's negative. Or maybe just a little weakly positive. Your whole algorithm is now shot. It looks like a staph, but it's acting like a strep. What do you do with that? This table just lays out the confusion so perfectly. You can see Staphylococcus saccharolyticus shares its morphology with a typical Staphylococcus epidermidis, but its oxygen needs and its catalase reaction, well, those look a lot more like some streptococci. And hey, pay attention to that intrinsic resistance to metronidazole. We're definitely going to circle back to that, because it's another huge clue that sets it apart. It's just a brilliant illustration of why it's such a master of disguise. And this all brings us to the most crucial point. This isn't just about a bug that's tough to ID. It's totally unique physiology. That slow anaerobic growth and the negative catalase, it's made it practically invisible to routine lab workflows for decades. And that means we've been massively underestimating how common it is and how important it can be. So why is it so picky? Well, the answer is written in its genome. Staphylococcus saccharolyticus is a classic example of what we call reductive evolution. Basically, it lives on our skin, which is a super stable, nutrient-rich environment, and over time it's kind of decided, hey, I don't need to make all this stuff for myself anymore. Its genome is just littered with over 300 pseudogenes. These are broken, non-functional genes. All that gene decay means it can't make its own essential amino acids, which makes it an auxotroph. And this is the perfect link between genetics and the bench. That auxotrophy is the direct reason for its slow, fussy growth in the lab. Thankfully, we have much better tools now to get around these phenotypic problems. MALDI-TOF mass spec can give us a fast, reliable ID, but, and this is a huge but, the database it uses absolutely has to have high-quality, genomically verified reference spectra. For years, public databases had the wrong references, which just made things even more confusing. Of course, the gold standard is gene sequencing. The TOF gene specifically is much better at telling these coagulase-negative staph species apart than the classic 16S rRNA gene is. Okay, so we've unmasked the organism. Now, let's talk about why getting that ID right is so critical. Because this bug isn't just some lab curiosity. It can be a pretty formidable pathogen. And get this, here is a fact that is truly astonishing. For an organism we all thought was super rare, modern techniques that don't rely on culture have shown that Staphylococcus saccharolyticus is actually the third most abundant staph species on the skin of the upper back. It's been hiding in plain sight this whole time, a common bug living on us that we just couldn't grow. And because it's living right there on our skin, it's in the perfect position to cause trouble. When it gets a chance to get inside, it doesn't mess around with simple skin infections. No, it goes deep. The classic infections we see are severe, and they're almost always related to medical devices. Think prosthetic joint infections, endocarditis on prosthetic heart valves, bacteremia, and deep bone infections like spondylodiscitis. It really is a specialist in chronic hardware-associated disease.
I love this quote because it perfectly captures its whole strategy. Staphylococcus saccharolyticus doesn't have the big, aggressive toxins like Staphylococcus aureus. It's much more of a stealth pathogen. It forms biofilms, it sets up shop on a prosthetic joint, and it just triggers this low-grade but persistent inflammatory response that, over months or years, leads to pain, bone loss, and eventually failure of the implant. It instigates. It doesn't attack head-on. This unique pathogenic style, this stealth approach, presents some really unique treatment challenges. So let's dive into the crucial topic of antimicrobial management.
So this is a really critical point, a huge clinical takeaway. Staphylococcus saccharolyticus is intrinsically resistant to metronidazole. This is a massive therapeutic pitfall. Think about it. A doctor treating what they think is a deep anaerobic infection will very often start the patient on metronidazole empirically. If this organism is the cause, that therapy is guaranteed to fail from day one.
Now, when it comes to susceptibility testing, we hit another hurdle. There are no species-specific breakpoints for this bug. We have to take our results and interpret them using the general EUCAST guidelines for all coagulase-negative staphylococci. The cephophytin screen is what we use to predict methicillin resistance, and then we need to do MIC testing for key drugs like vancomycin. And super important, rifampicin should never, ever be used as a standalone therapy.
Now, if we look at treatment guidelines for prosthetic joint infections, there's actually a really strong consensus. Both the Dutch SWAB guidelines and the international IDSA guidelines point to the exact same core principle for methicillin-resistant cons infections where the implant is being kept in place. You need combination therapy with vancomycin and rifampicin. Rifampicin is the hero here because it's amazing at penetrating biofilms, which is the absolute hallmark of these infections.
All right, to wrap things up, let's quickly touch on treatment in special populations, and maybe most importantly, how we can stop these infections from happening in the first place. Treating kids always requires special consideration. Kids are not just little adults. Their bodies clear drugs much, much faster. That means they need higher, weight-based doses given more often, as you can see in this table for key antibiotics like vancomycin and linezolid. And doing therapeutic drug monitoring for vancomycin is especially critical in kids to make sure you're getting the levels right.
Now, this slide illustrates a key theme for the whole story. Preventing disease from Staphylococcus saccharolyticus really starts with detection. You can't control what you can't find. It starts with clinicians getting the right specimen. Tissue is always, always better than a swab, and using the proper anaerobic transport media to keep it alive. And then, crucially, there has to be communication. The clinician needs to talk to the lab and say, hey, hold these cultures longer, for at least 7 days, to give the slowpoke a chance to actually grow.
And that really brings us to the main conclusion here. Successfully diagnosing and managing these infections is not a one-person job. It absolutely requires a partnership. You need astute clinicians on one side who suspect a fastidious bug might be causing a chronic infection, and you need vigilant microbiologists on the other who are willing to go off script, hold plates longer, and use modern tech to finally unmask this great mimic.
So, I'll leave you with a question to think about. Consider your own lab's standard operating procedures. What happens when you get a gram-positive coccus in clusters from a sterile site, but it doesn't grow aerobically and it comes up catalase-negative? Do you have a pathway to investigate these organisms that just defy the neat and tidy algorithms?
And that's our deep dive into Staphylococcus saccharolyticus. If you found this useful, please subscribe for more explainers on all kinds of fascinating microbes. We would love to hear from you. So if you've ever come across this mimic in your lab, or if you just have questions, please leave them in the comments below. Thanks for watching.